Welcome to AS Enzymes. Enzymes are the biological catalysts that make life possible. Without them, the metabolic reactions required to sustain your cells, from DNA synthesis to respiration, would happen far too slowly. In this unit, we explore how these remarkable globular proteins work and how their precise three-dimensional shapes dictate their extreme specificity. This topic sits at the heart of the Cambridge syllabus. It is heavily tested across all three papers, bridging your knowledge of biological molecules with the complex cellular processes you will study next. We begin with the mode of action. You will master exactly how an enzyme lowers the activation energy of a reaction by forming a transient enzyme-substrate complex. Notice how the active site is a highly specific cavity. The substrate does not just bump into it, they have a complementary shape, much like a lock and key. We will evaluate both the lock and key and induced fit hypotheses to explain this precise molecular interaction, giving you the exact vocabulary the mark scheme demands. Next, we investigate the environment. You will learn to explain how variables like temperature and pH dictate how fast an enzyme works. We will trace macroscopic effects right back to the tertiary structure. For instance, increased heat means more kinetic energy and more successful collisions, but too much heat breaks hydrogen and ionic bonds. The enzyme denatures, losing its specific shape. Examiners love testing your ability to explain these curves, and you will learn to avoid common traps, like saying an enzyme dies. Remember, enzymes are molecules, so they denature. What happens when you add more substrate? We will plot the rate of reaction. Initially, the rate climbs steeply as more active sites are occupied, but eventually the enzyme becomes saturated, and the curve plateaus at the maximum rate, known as Vmax. You will also learn to derive the Michaelis-Menten constant, which measures the enzyme's affinity for its substrate. Mastering this single curve and knowing how to draw a tangent to find the initial rate is your key to top marks in data analysis. Finally, we tackle enzyme inhibition. How can we regulate, slow down, or stop these catalysts? You will distinguish between competitive inhibitors, which temporarily block the active site because they have a similar shape to the substrate, and non-competitive inhibitors. Non-competitive inhibitors bind to a separate allosteric site, distorting the active site so it is no longer complementary. You will learn how to identify which is which simply by looking at how they shift our rate curves. By the end of this unit, you will confidently link the molecular structure of enzymes to their biological function. You will calculate initial rates from practical data, identify key variables to standardize, and evaluate complex inhibition graphs. You are ready to begin.